All right. Sorry about that, everybody. We had a technical issue apparently. We are now in part 2 as opposed to part 1. See if everybody, anybody/everybody shows up. Give it a second here. I mean, at least we got through. We got through the important stuff that we need to do. How fishy would it be if it just cut out in the middle of the other stuff? Part 2. Let's go. Let's go. We are back, everybody. Thank you for putting up with. I don't know what that was. Might have been a server side thing, actually. I don't know what the last things we were talking about is. If anybody had a question or something that you posted, go right ahead. Insurrection against Eric. That could happen, I suppose. If you had a question and I didn't get to it before the other stream cut out, let me know. We need more channels in the Discord server. A frequent request that I am apathetic to at the current time. However, I will answer your question about keeping thorax porcellana. I got them for the first time a month and a half ago. A bunch of larger nymphs, and I think there was like one adult male. And I have to say, they're doing very well. I've been pampering them. Frequent mistings, full top ventilation, fresh fruit, and the whatnot. So I'm hoping my first litter will pop up by the end of the month. I'm excited to see the vampire phenomenon with them. They're very pretty roaches in their own right, even without the quirks of that. So we'll see how they stack up to other epilamprinae. I've got like a cut here or something. See how they stack up to other epilamprinae. Because again, Rhabdoblata rustica is annoying me right now, which I never thought I would say because usually they annoy me the other way by having lots of babies, and right now they're not having very many. But I think they will explode now that they're in the big enclosure. Actually, Epilamprinae species Borneo probably, or Kota Kinabalu, probably the best for. They'll take pretty high density and a little bit more neglect than the other Epilamprinae. Opisthopledia go back and forth for me, where they'll just be doing kind of awful, and then I'll just kind of change things up for them, and all of a sudden they'll just perk up and be a very productive culture for a couple of months. A lot of them, they actually come from temperate areas. I have no idea where our hobby stock comes from. It's been around for a long time, but thinking back on the last couple of years, this has been kind of par for the course, this sort of ebb and flow with the Opisthopledia. Orang Mango, what do you feed immature Sinusoides quadrispinosa? They're in my colony is in a 5.5 gallon tank, and I do a fruit fly bomb every once in a while. There's springtails, 2 or 3 different types of springtails in there, small silvers. Tropical, yeah. Tropical pinks. There might be some Entomobria unostrigata invading that I might need to do something about. And then also a friend who has a good colony of Libetioides. Again, I think I forget the one I've written down. I think I've written Libetoides a couple times, but the publications are kind of hard to find. And other than I think TJ sent them to me in Discord, but I think it's actually Libetioides, not Libetoides, Libetioides, ornata, still A at the end. Libetioides ornata, that she does very well with, and feeds the there's a fish food called like bug bites or something, and it's like ground up black soldier fly larvae and stuff. And I've been using that as a bonus supplement food when my fruit fly cultures are in between booms for a lot of my harvestmen, and they do really like it. Libidioides albolineata. Again, this entity has been through hell in the last 15 years, because first it was that the eastern U.S. harvestmen were Vonones ornatus, and then it was determined that ornatus was the entity with the spines from Florida. So then we were calling the ones from the eastern U.S. Vonones sei. Very recently, it was determined that they're not sei. Sei is a western species in like Oklahoma, Texas, without markings. So then they were Vonones albolineata. And then also, I think in that same sweep, they dissociated the genus from Vonones, or maybe gave an old synonym had priority when they split the genus, so then they became Libidioides albolineata. So it's been absolutely a fustercluck, as it's called, for this individual. Which I will comment on the website. Something else, a feature I'm really looking forward to adding to the site that I think will be immensely helpful to a lot of people as time goes on, is everything will develop a list of past names, and this will be focused mostly on past Latin names, as again, in the plant community, it's joked that they use common names because they're more stable than the Latin names. This is true to an extent, as we see in the Libidioides situation. But I will focus on listing out the previous names for, or alternative names for, the strains of everything. So that will be a situation there, and they won't be declined properly and all this stuff. It will be, again, the Eastern harvestman entity will have Vonones ornatus, Libidioides, Vonones sei, etc., listed somewhere on the page with the additional information, so that it helps people to keep track of things. And for things that I got and that weren't ID'd, or things that I got and that were then ID'd, so other things like the Ergala pilosa in terms of roaches, which is not actually Ergala pilosa, but people have been buying them as that for years. So I will be putting that information on the website so people have it available. Exotic Wilderness asking if the website overhaul will add pictures of each enclosure of the species. That is on the to-do list. I would like to have nice, pretty pictures of enclosures for people. So it's pending enclosures that are in that state. For example, I will not take a picture of the Blatella germanica V5101 in its current state, where the barrier is degraded and the pine cones that I have in with them are smothered in roach poop and pyralis moth tunnels. When I refresh them, then I will show the image of the enclosure, and this serves two purposes. One, so people see what it looks like in its optimum state, and two, it will sell better. It is multi-pronged to show enclosures that are in good shape, obviously. And obviously in the Roach Care Guide and on the pages, it has some advice for keeping things clean and tidy and whatnot, which is harder when you have 500 plus cultures of things, but very doable on a small scale. I think back to when I had my stack of like 15 roaches, and I thought it was so much work to take care of them, and now it's like, man, you have no idea. Something says, I saw again my Lucky Hormetica nymphs using mouth parts on the back of an adult. So I recorded that behavior in Lamproblata. I think that it's in some species, it's probably, what's that called? Allo grooming. I think in some species, it's the bare bones beginning of social behaviors. Lucky Hormetica may be on the end of being a social behavior. So it's hard to sort of prove some of those things, because you don't want to go like that one publication a couple of years ago for the, I can't remember what species of roach it was. It was the shiny red and green roach from the Amazon. They did that whole publication where we found it's a eusocial species, and we found a queen, and we found them doing all this weird behavior with the queen, and it was white, and they were doing all this, and then every single cockroach keeper And also entomologists looked at that publication and just ripped it to shreds because it was completely vacuous in understanding of what was going on. And obviously it was determined that it was a teneral individual, probably an adult female, and the frenzy and tending to and whatnot that was being seen was the adult males trying to mate with her. It's common in some species of roaches for adult males to mate with just out of the exoskeleton matured females. And in some species, there's actually a fitness benefit to this. In Diploptera punctata, the male mating with the female immediately after she molts is because the glands that produce the defensive secretions have not begun working yet. And so, by the male being present with a freshly molted female, not only is he obviously getting dibs on her first batch of offspring, but is also protecting her from predators because the adult male is fully sclerotized and has the protective chemicals ready to go. So that's the advantage there. So that's what was probably going on most likely. So anyways, yeah, so that's interesting. I've seen that again in Lamproblata. I would say it's a bit of social behavior. Roaches obviously perceive their surroundings with their antennae and their palps, and so just checking to see if the thing next to that roach was food or not, or if it was another roach, something the nymph could have been doing. But there's something to it for sure. It happens enough across a wide variety of roaches that there's something going on there. Luch Garden State Bugs and Pods. Hello. Luch, I don't think I responded to you in Discord. Maybe I did, but I definitely still want those Percellio spatulatus. Chain says new stream who dis. Orang Mango asking if I have Dalquestia formosa. Let's see what that is. Oh, I wish. That's the one that TJ's always moaning about. I'm moaning about it too. I've never actually seen them in the wild either. Surprisingly. Rocky Woodlands is the habitat. I'm just reading off the bug guide page here. Seasonality is kind of all year. Maybe they might even be a cool weather thing. That's a cool genus. Dalquestia. We're finding, TJ's finding with a lot of cosmetids, a lot of harvestmen, they are inspired to oviposit by cooling the culture. So I don't think this is the case with Sinertoides. It's a tropical species, and I have not seen evidence that they will stop laying if they're kept warm for a long period of time. But with Libidioides sayi and Libidioides abulineata, it seems to be the case that if you end up with adults and no babies for a while, you can trigger the adults to start laying again by cooling the culture. So that's just some information I have in your back pocket there. And that does not seem to be the case with Ornatus either, that they do not need to be cooled in that way. But Eumesosoma is another one. So Brain asks, will the new site have live video feeds for every enclosure with temperature and humidity readings? I can hope I get to that in 30 years. That would be pretty neat. Meloroidea, I think that was the genus name. Alan adding some more information about that. The bogus, it was published. I can't say it's bogus, but obviously everybody's ripping it apart. They may have retracted it, or maybe they just left it up for some poor sap to read through and become infatuated with roaches and being eusocial while forgetting about the fact that termites exist, I guess. Speaking of termites, I think they were reticulotermes from Foster's, Alabama. I put a polycarb over the whole lid. No faunarium lid or anything like that. Just slapped a polycarb on top of it. And I've destroyed their Tower of Babel a couple of times now. And they stopped forming, and I thought, oh, that's interesting. So then I looked a little bit closer at the water port, and it turns out that they have excavated and presumably eaten the hot glue that I used to secure the watering port to the front of the enclosure. And I'm not too thrilled about that. I'm going to keep an eye on them and make sure they don't start advancing forward more. I assume they'll start piling up some poop if they start moving out of the enclosure to make like the scaffolding to leave. But definitely in another realm. Those guys are in terms of eusocial insects. You get the eusociality, you get a little bit more. You get what's the swarm intelligence going on there. Swarm architecture, I guess maybe you could call it. They start thinking about not living where they currently are. Meloroidia magnifica. Some eggs and Allen have pieced together the name. Tennyson says, hello. Tennyson, after the holidays, I need to get a restart of the Pseudophilus from you. I think mine are done for sure this time around. Satchel asking if people have erginulus in culture. Let me check that out. Actually, speaking of gynulus and whatnot, I got something to add. I don't think so. There was a big Harvestman import years ago, and nothing really ever came of it. I know they basically got a target for March. I didn't know there was a. Let me look that up. See if there's a member of the genus in the United States. One record from Florida. I thought Alan was keeping those. Maybe. Sorry if I just exposed you, Alan. Some of that stuff is some of these things are just fumbling around in South Florida, and then you come across something under a board, and all of a sudden it's hype. Satchel, I'm not sure if you're aware of Sinertoides. Those are Erginulus are pretty cool. I'm not sure if you're aware of Sinertoides, Quadraspinosa, but that's kind of the current, fairly readily available, cool cosmeted harvestman from South Florida. Stignama, Alan says, is the species he keeps. It Stignama. Yeah, maybe the Erginulus will be something we may just come across while crawling around. But yeah, Sinertoides. Check out Sinertoides if you weren't aware of them yet, Satchel. They're basically everything people want from Says and ornate harvestmen and whatnot, but bigger and kind of betterer. Yeah, I'm aware. All right, just wanted to make sure. It would be nice if we got some more harvestmen that weren't just sort of like orangish reddish brown. It's a nice color, but it would be cool to get some color diversity. Paleata are doing great. All life stages are everywhere. See you later, some eggs. Thanks for joining us. I was gonna go on a little fun aside here on a fun genus of slugs, Vaginulus, which some of our pancake slugs used to be in. A person wants to culture Ortholasma. Let me see what those guys are. Oh yeah, that's pretty cool. Ortholasma rugosum was a West Coast thing. Yeah, of course it's a West Coast thing where all the fun stuff is of this sort. All the weird stuff that looks like pebbles and stones and has had millions more years of evolution to turn into all kinds of crazy stuff. One day I'll do a big thorough Cali trip again. Same with Texas and Puerto Rico. Satchel says, apparently the black leatherleafs are established here in Tuscaloosa. Yes, that is the species I was going to bring up. Used to be in the genus Vaginulus, now is Bilocaulus, I think, Bilocaulus angustipes. I was down to one, I think maybe even the unfortunate auction person who won the group, not this last auction, but the year before, or maybe it was the spring auction. I thought mine were doing really well, and then poof, I was down to like none. And I actually found a dead one that had just squirted out the last of its eggs, and I thought, okay, well, let's try and make this work. And the eggs actually did end up hatching, so I was happy about that. And then I ended up rearing only one to adulthood. I figured, well, they're hermaphrodites. You can probably self-fertilize. And I was right. It did self-fertilize, and I have a colony of them going again right behind me. But Jojo and I had a lot of fun because that one individual has gotten titanic. It's the biggest Bilocaulus I have ever seen. It's like Florida leatherleaf size. And we did a whole nice photo shoot trying to get it to come out and to have the eyes emerged from the mantle and all that stuff. It was one of the little mini highlights. Took quite a while. It looked like a dog turd on a plate for some time, but we got there eventually. It was good laughs. Yes, Jojo, it was good laughs. So yeah, Belocalis. And I think I'm down somehow in my Florida leatherleafs to just one individual too. I don't know how. I have been. Did I bring this up in the last live stream? I'm not sure if I did. I've been paying more attention to substrate acidity in chemistry, and have noticed that as substrates go more and more acidic, a lot of things don't do well. Makes sense. You think of the boreal pine forests, extremely acidic, peaty soil. What's that stuff called? It's in Minecraft. What's that stuff called? They call it something, and it's Podzol. Podzol, I think it is. A very acidic soil has a lot of organic matter in it, so you think that Podzol. Thank you, gamers. So you think there'd be a lot of stuff in it, but there's usually not as much as you would think. It's usually more fungal and bacterial. It's a very complex relationship between those things and plants, not necessarily the best for invertebrates to be involved, or if they are, it's very specialized. So I've been watching substrate acidity and trying to see if there's a correlation between substrates going more acidic due to fungal activity or just due to waste accumulation and cultures crashing of various things. Not so much roaches, but isopods, millipedes, springtails, other stuff. So I've been keeping an eye on that, and I'm thinking that was brought to my attention. Somebody here, I don't want to dox anybody, and I'm trying to think of their name, but who got some snails from me to start offering more calcium to them. And surprisingly, the snails one of the snails did not want anything to do with the calcium that I had offered. The others, the Neohelix, went right for it. So I'm working on modifying the pH of the soil using different pH altering supplements. So we'll see how that works out. But I dumped a bunch of pulverized lime in with the Belocalis, and it seems to have helped. Even though they're slugs, they still do need calcium. Definitely help with the Neohelix too. But I'm just sort of watching how that goes, because I really want to save that culture of Florida mantle slugs. And I think I might be down to one. Again, I was feeding leafy greens. There's actually some sweet potato vines just growing in there too. And it's been shown that they will eat high quality food, and they can persist on low quality food like leaf litter and rotting wood. If you really want to get growth and reproduction, you have to give them leafy greens and higher quality food like dog food and whatnot. But I was offering that and not getting good results, so now I'm eyeballing the soil pH. While at the same time, there's a huge colony of rubber ducky isopods in that enclosure too. But I sell out of it pretty frequently, so I don't think that's causing the declines. It's still finding eggs and stuff. It's not like the isopods are eating the slug eggs. So Ray Manga was taking a sneaky peek at the Big Pine Key on your website. Are they easy? For the Porcellanites virgatus, that is definitely going to change when molecular work is done. Those things are a world different, and in a world different environment than the Porcellanites up in Louisiana. That is a big old species complex that needs to be cut into pieces. Anyways, but yes, those should be on there. Satchel says, what do you think about the possibility of there being like 5 different leatherleaf species in Florida? I find it invigorating, and I bet that we would find the introduced ones in the more disturbed habitats. Zen Monkey says, so all of a sudden those crazy ice pie keepers with all their limestone makes more sense. Yeah, but not a brick of limestone, more of a soil additive to moderate the pH, if anything. But also taking into account that I've kept several species like white tigers, white duckies, and rubber duckies with no supplements whatsoever for 2023 been almost 6 years for the rubber duckies, and they've been thriving. I've never run out of them since I've had them, and sell a good number every month. So it's perhaps one piece in a much larger puzzle of things. So it's possible that maybe certain fungi in the soil or bacteria will start to turn the substrate more rancid or alter the pH that then makes it not conducive for isopods to survive, or millipedes or what have you. And then by doing a very abrupt turnaround of the soil pH, you change the bacterial or fungal composition of the enclosure and you rectify things. So Shane says, Meloroidea af magnifica is that eusocial roach, still beautiful, and I would love to get them. Yes, it does not make me want them any less that they are not eusocial. They clearly have some behaviors around accumulating, congregating, much like Durapeltis do. So I want them still. Satchel says, I got some of the calcium chips that they make for aquatic inverts. That's just like calcium and spirulina, and giving them to my terrestrial snails, and they've been a hit. Yes, definitely for snails needing lots of calcium, and using the pulverized lime is a quick and easy way to give it to them. I guess cuttlebones too, but cuttlebones are more expensive. So, nope, limestone brick right on top of those ice pods. Yes, the most effective strategy is you get a big pane of limestone for $150 shipped. You put all your isopods on top of it. You get another pane for $150 shipped, and then you just smash the two of them together, and that's proven to be the most effective. It gives an immediate, gigantic delivery of calcium or lime or whatever directly to the internal organs of the isopods, and they love it for a very short time. Several of my isopods just demolish cuttlebone. So yeah, it's I've seen them eat plaster of Paris too, which has that's not calcium. Was it manganese or magnesium? Plaster of Paris. Let's see. Gypsum. Let's see. Gypsum is calcium sulfate dihydrate, but is the calcium in is it in an accessible form that the isopods can use is the question, because some of those bonds are not easily broken. Curious what exactly plaster of Paris has. Gypsum hardens when moistened and allowed to dry. I mean, I've seen them. Or some other things. Limestone isopods, Samichi. I'm trying to think what DAP white plaster Paris. That's what I have. Let's see. White plaster of Paris ingredients. It's also with millipedes, with pill millipedes. A lot of them are reported as being found on more calcareous or limey soils. So I'm curious. It's probably a combination of things with them having poor mouth parts and also perhaps needing more calcium than other millipedes or than other invertebrates do. Trying to see what find the exact composition of the plaster of Paris. So it's what's this percent of chemical in preparation? So this is plaster of Paris as an ingredient, but I can't find out what exactly plaster of Paris is made of. Apparently, it varies by region what they actually use in the mixture. Cement sometimes too, gypsum, lime. So I may just have a heavy component of lime, which is a type of plaster of sand, water, and lime. It's a lot of apparently a lot of things. You need to be a chemist to find out the exact measurements of things. Exact willingness asking about how I'm keeping my Eurycotis decipiens. I'm pulling them out of a slump right now, so I set them up in a 10-gallon with a lot of just decadence, all flavors of rotting wood and a little bit of leaf litter and some pine cones. They're pretty low density right now. And again, I'm trying to pull them out of a slump they were in. I let them get very crowded, and I had flower pot fungus. Somebody probably remembers me on one of these live streams saying that I needed to redo my Eurycotis tachypians because the substrate was being taken over by flower pot fungus. And that is a fungus that definitely makes the soil, or makes the substrate inhospitable to a lot of organisms. Not everything. Anadenobolus ocraceus seem to like it. Makes sense. They're from more tropical areas where maybe that fungus is found in the wild and naturally occurring, or a relative of it. Anadenobolus ocraceus don't seem to mind it. A lot of larger roaches don't seem to mind it, like Blabberus and Blabberus, Eublabberus, Blaptica, hissers even. But for smaller stuff, it seems to, again, it might alter the soil pH and make it way too acidic or something like that. So mine are on my sterilized outdoor compost now with the max accommodations, because it's one of my favorite genre, and I need to pull them out of that slump because I really like them. So, Eric Matthews says, no telling what bugs get out of the odd things they crave when mealworms eat styrofoam. Yeah, the other thing is with stuff like millipedes, and it probably applies to some degree to some isopods, is with millipedes, large spirobolids. I'd have to ask my friend Mike which species was mentioned in the publication, but they act as biomagnification chambers for generalist bacteria in their surroundings. So they'll just go eating through the leaf litter. They'll pick up different bacteria that are just in the leaf litter, and then provide just the perfect mixture of moisture and chemistry and whatnot to get those bacteria up to very high numbers, so they can break down the substrate faster. So the flip side of that is, now imagine they are eating substrate that contains bacteria that are not favorable to them. Whether that's because a bunch of millipedes in the enclosure died because of lack of feeding, or dry, or too wet, or something. And then those bacteria seep out of the corpse into the substrate, or even just the millipede eats it nibbles on the other dead millipede or something. Then you get this sort of domino effect of more bad bacteria that are incompatible with the millipedes amplifying, and all of a sudden your culture of them dies. So some of it's what bacteria things have going in their condition. You can kill a culture of lobster roaches with bad conditions, or you can if the culture's got good momentum and is doing fine and going really hard, you can feed them pretty much anything and they're fine. So there's the history of the culture that really needs to be taken into account with a lot of things. So a person says, I found some Tylobolus in a pile of limestone, wood, and garbage the other day. I think a lot of stuff can benefit from it, not necessarily required though. So soil pH probably matters, as well as the bacterial composition of what you're feeding them. Stephen Logan, this is part 2 because part 1 crashed, so I had to start a part 2. Did you ever get the labia minor? I unfortunately did not get the labia minor. That contact, her box of stuff arrived in good shape, and hopefully they can be found in the future, as I still do really want labia minor. If you're offering labia minor, if you found some labia minor, let me know, because I do still want them and still need them. Alan says, in my experience, mycelium of that fungus makes the substrate inedible, so millipedes and isopods seem to starve. Also, their spores stick to inverts easily, and that seems to stress them out. Thank you, Alan, for weighing in on that. There's another fungus. Again, I should send it to my friend Jules to ID. She's been on a roll with fungus ID and whatnot lately. There's another one. It's not flower pot fungus. It's not a pleurotus. It's not inky caps, but it has just your standard old brown-looking mushroom that's popped up in a couple of bins. And a few times I've seen Narcissus americanus crawling up onto it and eating the mushroom itself. It's not the same thing as the mycelium, so the mycelium may not be good, but they were at least eating the mushroom, or perhaps they were eating the mushroom because they didn't have anything else to eat. So, and then also, Alan, there's that other weird fungus which has spread to a couple of my enclosures now. I'm only just a little bit worried because it's still pretty isolated, and without a fruiting body, it's easier to manage. But there's that dusty gray mycelium that one of your enclosures had that has popped up in a few. Excuse me, a few of mine. Just keeping an eye on it. Again, it's not a problem. I'm just making sure it doesn't become a problem. Eric Matthews says, "Oops, on the flower pot fungus, my millipedes are getting a lot in their tank now." Yeah, it seems to be. Again, some stuff doesn't seem to mind it or likes it. Anadenobolus ocraceus seem to like it. Spirobolellus seem okay with it, and Leptogynaeolus sororinus are okay with it. It seems. How big? Paniolus. The the head is kind of maybe like 2-ish inches across. That seems like a reasonable match, actually. And the spores are almost the exact same color as the head. It's like a dusty tan spore that it leaves everywhere. And the gills are pretty evenly distributed. I don't know if they attach to the stem or not. I can't remember. So, but yeah, so that's the inner ecology of our enclosures is something to always take into account. I should also say my white tigers, which had a crash, I think I removed them from a millipede bin. Again, using millipedes to facilitate isopods is something I'm a big fan of. And some of my biggest thriving isopod and millipede cultures are co-housed. But my white tigers, which I removed from a millipede enclosure, then crashed. I was left with one male. I was kindly sent a female as a gift from Alan, and I am finally getting my first set. I just got my first set of babies from them during the photo shoot, actually. I took the male out so that we could get a picture. Jojo could get a picture, but I left the female because she was with child still. They were all underneath her. So the colony is on the rebound, and the males from stock that threw whiteouts. So I'm hoping that those will pop up and I'll get them isolated. Chain has had the Paniolus, pretty common in mulch, and I've had them pop up a few times. Okay, yeah, that could be it. I'll have to get an individual picture of one to send to you, Chain, along with that mystery weird veiny mycelium that I found from Huntsville, the Huntsville millipedes. I have failed with Chalktella cummins. Did not get a colony established. I had gotten some babies sparingly initially, but never got anything more, and I gutted the colony, found no living adults. My Chalktella huberti are still alive, and I'm going to bump them into a new enclosure, but only like 5 or 6 of them are still alive. But I'm going to give cummins another go this spring, and I think that having seen them in the wild twice now, I think the key is they just want high humidity, like sky high humidity. So people who have gone after Huberti have said that by the heat of the not just the heat of by the dryness and heat of the summer, they're gone from being under logs and in leaf litter. They just disappear. I have not seen cummins habitat during the summer, but having seen it during the winter/spring, where they're extremely abundant in leaf litter, in logs. You see piles of poop when they're next to them, when they've got a bunch of leaf litter clumped up next to them. They've clearly been eating the leaf litter. You see them on the underside of the well-rotted logs eating it. But that area is very moist. There is a spring that runs through that area, and you can find them within like 150 feet of that spring. And then as you sort of go away from it, they seem to be replaced by, or the composition of millipedes changes, and Narcissus americanus seem to become a little bit more common. So that might be, they might just want really high humidity. It would also explain their limited distribution, that they have very low dispersal beyond these humid enclosures. So I will give cummins another go. I have an enclosure set up for them, and on top of that, I've taken some of that mycelium from Huntsville and inoculated the new enclosure with it. It seems to be a very easy to care for strain of whatever it is. It just infests the hardwood pellets that I rehydrate. It does so pretty quickly, and millipedes seem to love it. I'm convinced it's the key to, in addition to the humidity, it's key to breeding the was it Sigmoria latior? I think it was Sigmoria latior. Yeah, I'm convinced it's key to breeding the Sigmoria latior. Is it breaks down the wood to just the perfect palatable state, and if you keep the humidity high, a lot of these flat millipedes do not have any sort of dryness tolerance. Uriuridi is like that too. When I had success with them with high ventilation, it was they were in the humid microclimate of the lower layers of the substrate. So basically, there was not airflow out of that area. It was its own separate mini thing beneath the drier, hardened over layer at the top. So I'm reapproaching how I take care of flat millipedes now. Still with some ventilation, though. Still with some ventilation. Because completely restricted airflow is not a good idea, especially if you have active mycelium. Keep in mind that the mycelium is using oxygen as well as the millipedes. And it may not be a lot of millipedes, but when 1/5 of the volume of the enclosure is mycelium, and there's also millipedes in there breathing, you're going to need some give with oxygen. Anyway, so that's just a couple of things that I am reapproaching with millipedes. Also switching over to leaf litter for Chicobolus over a lot of rotting wood. I've had success with rotting wood, but I've had more success from the enclosure, more consistent success in enclosures that were fed well-rotted leaf litter, the kind where you pull it apart and it just kind of doesn't crinkle, it just kind of pops apart or kind of tears apart. So I'm that's another thing that I'm looking into, and hopefully it will be an exciting next couple of years for millipedes all around. To those who are still waiting on your was it purple or pink dragon millipedes, the main culture is gone into a lull of production. I see a handful of sizable babies, but nothing like last spring when there were just piles and piles of them. So I've moved all the adults to a new enclosure, decreased the ventilation, chalked it out with what I'm pretty sure what I used last spring to get the colony going, but we'll see. We'll see how things go over time. And millipedes are the best. Yes, Luch, they're very nice. I do really love the millipedes. They get to fall in the honorary cockroach category for me, I think. But still a lot we have to learn to get consistent results out of them, and I'm really hoping to make good on the next blob of millipedes that somehow gets into the country that Underground Reptiles and random expo vendors have available. Was this not the Spirobolus? There were a lot of the the Southeast Asian species that popped up. I'm ready to capitalize on that and try and get some of those going more of an earnest effort than I have before. Again, I've even got some enclosures prepped up and waiting. And final thing, we're at 43 minutes, 8:30. This has been one of the longest streams. Thanks everybody for tuning in for so long as we keep going. Pergodesmity, the little, I don't think they have a common name. We have two species in culture. We have one from the Kota Kinabalu from Borneo, I think Borneo. And I accidentally started culturing a species from Auburn, Alabama that I collected, or maybe Alan collected with the pill millipedes, which are all still alive. I just haven't gotten any reproduction out of them. But I have prepared an exuberant enclosure to move them into that I'm hoping to get good results out of with this particular set, the Auburn species or genus. So yes, I'm going to move the Pergodesmidae. The Kota Kinabalus are doing excellent in with the Benuatulus species Cowsock. I think that's how you pronounce it. If anybody wants to correct me, go ahead. So I'm going to move the I'm going to move the Auburn Pergodesmidae in with something, maybe in with a Narcissus or a Chicobulus or something. I'm going to pick something and just throw them in, hope for the best. But I do like them. I'm liking these tiny millipedes. I don't know if I'll ever bring myself to keep Julids. There's a couple of big introduced Julids. I might dabble in those eventually if I find a good group of them. I would be hard pressed to keep Oxidus gracilis too. Would be very hard pressed to keep that species. Helicorthomorpha will be collecting on the Florida trip. Definite target. Should be an easy thing to get going again. So a person says accidental cultures are the best cultures. Yes, love when something accidentally cultures. And Chain says, tiny beautiful millipedes. I will start my colony next year. Yes, Chain. I have plenty of the Pergodesmids from Kota Kinabalu. I'm happy they are going up on the site. Everything is going up on the site. Things that do not have pictures taken yet will be going up on the site. We'll be getting pictures added during JoJo and I's next photo shoot, which we're eyeballing April, but we'll see if we survive the winter and the auction and all the other things. Eric Matthews says, I need a video on how to get more efficient on hydrating and feeding my enclosures, especially after this raffle. You can do the front watering approach. These guys are good to go for another 2 to 3 weeks at least. And depends on what species and all that other jazz. But yes, optimization is very important with large collections. And also, as I find out more and more over time, having good substrate on hand is important. But I'm good on roaches, good with roaches and substrate because the compost pile has been ever yielding. I need Polydesmus again, or Pseudopolydesmus. I had forgotten about that. I tried to collect them in spring after my colony failed, but no such luck. I didn't get enough of them, of the Pseudopolydesmus at one of the sites we went to. I think it was in Buell, or maybe it was Foster's. I didn't get enough of them, unfortunately. So I need to get Pseudopolydesmus going again. You have Serratus. Well, let me know, Chain. Keep me posted. Text me or whatever, wherever you have, and we can go from there. I'm happy to trade some Pergodesmids and other MISC stuff that I have for other things. So, okay, guys, we've had a long collective stream. It was good to be back and check in with everybody. And I look forward to getting everybody up to getting the website up and going. It's happening. I gotta get past this expo this weekend, and it's going to happen. JoJo took some absolutely beautiful pictures of springtails, beautiful pictures of roaches, beautiful pictures of pseudoscorpions, all kinds of things, as much as we could get done. And I'm excited to share all of that with you guys and share the knowledge and have everything nice and organized. If we can get it in the direction I'm hoping for, it will truly be an asset that I hope the people will use for years to come. So take care, everybody. And until next time, may your cultures ever be bountiful. Thank you again to the donation drawing winners and whatnot. JoJo's only gotten through 10% of the pictures. So share screen next live stream. I'm not sure what that means. But anyways, take care, everybody. I'll see you around. May your cultures be ever bountiful, etc., and all that good stuff.